Alright, welcome back, Biology B students. Sorry about the quick abruption. I forgot that these videos are only 15 minutes long. So we were moving our way back into the test cross. Now, when we cross with a homozygous recessive individual, all that this illustration is taking a look at is if we're looking at an individual that is heterozygous, let's take a look. Let's look at this one at being homozygous dominant. That homozygous dominant, if I have that one parent that's homozygous dominant and I cross it with one that's heterozygous, we know that this guy can only give a big S, this guy can only give a little s. So that means all of our offspring, our progeny, are going to be heterozygous, which means that they're all smooth seeds. Now, if I use this guy as the parent, this guy here, this guy can either give a big or it can give a little. This guy can only give little. So what I have here in my test cross is I can get half of them smooth, half of them wrinkled, and that is what this is corresponding to over here. Okay, if I do homozygous dominant cross with a homozygous recessive, 100% are still going to be dominant trait. But if I do a cross between heterozygous and homozygous recessive, half are dominant, half are recessive. Alright, let's skip back along to looking at what's now considered a dihybrid cross. So now not only am I looking at one trait, now I want to take a look at two individual traits. So in this case, we're going to not only look at the smooth shape versus the wrinkled shape, but now we're going to do it in terms of color or not. Let's represent capital Y is dominant for yellow. So if I'm looking at setting myself up with kind of a little legend or a key, capital S is smooth, okay, lowercase s is wrinkled, capital Y, yellow, little y is green. That's just giving us a little legend of what we're looking at in terms of our alleles, our traits that we get from mom or dad. Now, in terms of our picture, if I want to look at this picture a little bit, I want to just blow it up a little bit for us so we can see. We have our cross between a smooth yellow. Now, here I'm just giving you the illustration that it's homozygous dominant. This could also be heterozygous for that trait. It could be two big S's or big S, little s. This is also the homozygous dominant for yellow. Now again, being that it's a dominant trait, it could be homozygous or heterozygous, but still show the yellow color. But in our instance, what we're looking at, let's just be very simplistic, is that we're both going to be homozygous dominant traits. I want to find the possible gamete combinations for that. And what I've done is I've shown them here. Over on the other side, if I've got wrinkled, the only way I can be wrinkled is if two little s's. The only way I can be green is if two little y's. I can only show that combination. Now you're thinking to yourself, well, how the heck did he find that combination? What you do is just, let's kind of just move this off to the side a little bit. The quickest and easiest way to do it is say, all right, if I have an S and two big Y's, my combination, I cross them 100% of the time, you're going to get this all the way through. And that's what's referring to those guys there. Same thing is concerned if I have little and little. 100% of the time, you're going to have the two little sections, which are shown there. Okay? Where it does get tricky is that if we start to look, let's kind of move this off to the side a little bit. If we start to look and say that this guy was actually heterozygous, so mom or dad was this, that's where it starts to get a little tricky. You can do the same thing. So we can still do this and find out our possible gamete combinations. Let's just keep the S's in the beginning. Those are the four different possible gamete combinations. Now remember, when a cell undergoes meiosis, how many cell combinations do you get? How many daughter cells are ending? You should have four. 1, 2, 3, 4. Four different possible combinations. So when looking at this, this is what my possible combinations would be. Another way you can do that is just what in math you do, kind of the FOIL method. We're going to take this big S with the big Y, and we're going to take this big S with the little y. So we have big S, big Y is one combination, big S, little y is a second. Then I can move my way into the little s, little y, little s, big Y, I should say, and then the little. I have the FOIL method. Those are my four combinations, which are also shown there. Those are the four combinations if in consideration it was heterozygous for our traits in terms of our genotypes. So as we move our way on, it looks very, very confusing. What this is just taking a look at two individuals that are heterozygous for both traits. Okay? What we're going to get, and you're going to want to know this and jot it down somewhere, is that if you cross two heterozygous individuals, you are always going to get that ratio. What that ratio refers to is its phenotype. Now think to yourself again, what does that mean? Physical appearance. Okay? 9 of them, let's kind of zoom our way around here quick, are 9 to 3 to 3 to 1. 9, 3, 3, 1. 9 of them are going to be dominant for both. What this refers to is both traits. In our case, it's going to be dominant for yellow and smooth. 3 of them are dominant for 1. Erase that. Dominant for 1, recessive for second. Okay, this one is going to be recessive for 1, dominant for second, and this one is recessive for both. So in our cross, we only have one chance out of actually 16 chances in which we can get wrinkled and green seeds, recessive traits for both. Now, in terms of writing that out, let's give ourselves kind of our own little square. I'm going to grab ourselves a new slide quick. And if we look at a Punnett square, we're drawing one out. Okay, now that we have this one drawn out, we're going to take a look at our combinations. Now, remember from the slide we had before, we already figured out what our possible combinations were. We had one big S, one big Y, one big S, one little y, one little s, one big Y, and one little. That is if I have this as my one, and if I'm crossing it with another one that had the same type, we're going to have that same type of gamete combinations on the other side. Now, in going through and doing that, you can go through and fill out all of these guys. Big, 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 all the way around. Okay? You fill out all these guys, you fill them all the way full, and what you're going to end up finding is in this big picture over here. Let's kind of zoom back in on it, and we should be able to kind of visualize what's happening here. Okay, let's really zoom in on here in our 9 to 3 to 3 to 1 ratio. Okay? 9 to 3 to 3 to 1. What that refers to, 9 of them are going to have yellow and smooth, dominant phenotypes for both. So yellow and smooth. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. 3 of them should be smooth but green. 1, 2, 3. 3 of them should be yellow, the dominant yellow color, but they're wrinkled. 1, 2, 3. And then you only have the one chance, so a 9 to 3 to 3 to 1 chance of actually being green and wrinkled. And just to finally end, so this is just kind of taking a look at that same thing, it's just what we just went through and did. So we're going to skip over that. It's just talking about Mendel's and the laws of independent assortment. Now remember, we just got done with meiosis. We talked about how certain genes, the two members of the gene pair, separate from each other during the formation of gametes. You don't know when they separate out during meiosis. These genes are going to be separated. We're not sure which you get. And so in terms of this sperm and this egg, this may be what dad could give because based upon his mom or dad, but you don't know if he gives you the big one or if he gives you the little one. Same with mom. You don't know if she gives you a big one or a little one. If they're both big, you know they have to be a big. If they're both little, they could be a little. Okay? So that's in terms of the gene segregation. In terms of independent assortment, all this is referring to is that when they do start to separate out, they separate out independently from one another. They don't need to be tied together. The combinations, you guys remember getting their hug on after anaphase, in prophase 1, when they get their hug on, they start the crossing over, and then they move themselves to separate out, and they're in metaphase 1, and then the whole chromosome gets pulled apart. But some of the genetic material at this point is now variable, and it's being that it varies, it's different, and some of the genetic combinations within those can all vary. Okay, don't need to pull it pulled together, and so that's what it talks about Mendel's law of independent, or principle of independent assortment. Now that's going to be kind of completing our notes. We'll take a look more at some notes when we get back together in class and finalize some other key vocabulary. Hope you guys have a good rest of your day.